Right, the first part of Chapter 4, which deals with the internal and external anatomy of prokaryotic and eukaryotic cells, we're going to focus on comparing and contrasting prokaryotic and eukaryotic cells. Okay, we're going to look at identifying shapes and arrangements of bacterial cells, and we're going to compare and contrast the structure and function of gram-positive versus gram-negative cell walls. That's going to be very important when we look at the gram stain procedure, which maybe we'll get to by the end of next week. So first of all, looking at the terms prokaryote, eukaryote. Pro means before or pre, and karyote deals with nucleus. So pre-nucleus. Eukaryote, eu means true, and of course karyote means nucleus again. So eukaryote means true nucleus. So prokaryotic cells do not have a nucleus. Eukaryotic cells do have a membrane-bound nucleus that contains the genetic material. So let's kind of contrast these two here. Prokaryotes, the DNA is not enclosed in a membrane, meaning there's no nucleus. Instead, there's an area called a nucleoid that contains the genetic material, whereas in eukaryotes, it is in a membrane-bound nucleus. Prokaryotes have a single circular chromosome. And if you think of eukaryotes like us, we have multiple paired chromosomes that are more linear in shape. Prokaryotes do not have histones, whereas in eukaryotes, the DNA is wrapped around special proteins called histones, and these play a role in regulating gene expression, which genes are going to get turned on, which ones are going to get turned off. There are no membrane-bound organelles in prokaryotes. I should add that, membrane-bound, whereas there are membrane-bound organelles, because you're going to see that there are ribosomes in both prokaryotes and eukaryotes. They just don't have a membrane around them. Prokaryotes have peptidoglycan in their cell walls, whereas eukaryotes, if they have a cell wall, such as plants and fungi, they have some sort of polysaccharide cell wall. Plants, remember, it's cellulose, and same with algae, and fungi, it's chitin. These reproduce by binary fission, and eukaryotes have cell division by mitosis. So some differences between prokaryotes and eukaryotes. Now, as far as the shapes, there are three basic shapes: bacillus, coccus, and spiral. Okay, so bacillus is rod-shaped, coccus is spherical or round, and spiral is some sort of twisted type. And so we can have spirillum, which are kind of corkscrew, vibrio, which are curved rods, and spirochetes. These are helical and flexible. Okay, and we have to be careful that if you see bacillus written in an italicized, then it's talking about the genus, like some of you have Bacillus cereus, Bacillus subtilis, or Bacillus megaterium. But it does indicate to you what shape you should be looking for when you do stain your bacteria, that it should have that rod shape to it. Now, bacteria can also have arrangements in addition to their shapes. So anytime they come in pairs, then we just add the prefix di, because di means two. So a diplococcus would have two round structures. A diplobacilli would have two rod-shaped ones connected. If they come in clusters, we use the term staphylococci. Okay, so they look pretty much like a cluster of grapes. That's what you'll look for. So if the name of your organism starts with Staphylococcus, it's a good indication that they're going to be round and in clusters. If something comes in chains, then we have the prefix strepto. Okay, so streptococcus could be coccus that are in chains, or you could have streptobacilli, which are chains of rods. Okay, we don't generally have a staphylobacillus. That doesn't really happen. Usually it's just a staphylococcus. Okay, now some other more unusual shapes is that sometimes they can come in tetrads. Okay, that's a group of 4. Sarcinae, which is more like a cube-like structure, in groups of 8. So there it has, instead of just the 2 planes, like a tetrad, it has 3 different planes along here that it can divide it through. And so you have all these different, packed into kind of like a cube. Coccobacilli is kind of an oval looking. And so it almost looks like a short rod or a plump, longer coccus, kind of oval shape. So usually they tend, if you had a bacterium that looked like this, you might classify it more as a coccus than a bacillus. If a bacteria is monomorphic, it maintains a single shape. And if it's pleomorphic, it means many shapes. Okay? So those are some different shapes and arrangements of various bacteria. So if we look at the actual structure of a prokaryotic cell, this is the diagram here that you should really learn and make sure you know, because it kind of takes us from the outer layers of the bacteria all the way to the inner layers. And it also shows you in kind of a cartoon form and then the real form off to the side. It says here that not all bacteria have all the structures shown. Structures labeled in red are found in all bacteria. So cytoplasm, ribosomes, a plasma membrane, just the same as saying in cell membrane, and then a nucleoid containing DNA. Okay, all of those are found in all bacteria. And then other bacteria can have a number of these other structures as well. So let's start on the outside and kind of work our way in. The outermost layer is what we call a glycocalyx, which means sugar coat. It's outside the cell wall, so it's even before we get to the cell wall. And there's 2 types. The capsule is more firm and organized, and it gets firmly attached to the cell wall. This one helps contribute to virulence of a bacterium, and it also prevents phagocytosis, which is kind of cell eating by like your white blood cells or things from your immune system. And so bacteria that are able to have a capsule as their glycocalyx can kind of sneak through your defenses sometimes and cause disease. The other type is what's called a slime layer. And just like the name suggests, it's more unorganized and loose. And so this one is a component of biofilms, and it protects against dehydration. And this one really aids in attachment. Really, when you look at all of the different both types of glycocalyx, they are helping in attachment because they're usually sticky. Okay. Flagella, again, microbes may or may not have a flagella, and you should know some of these terms to kind of describe where those flagella are located. If a bacterium is atrichous, that means there's no flagella. If it's paratrichous, it means that there are flagella covering the cell. And then if it's polar, it means you can have flagella at one or both ends of the cell. And so there we can get more specific and say monotrichous means that they have a single flagellum at one pole. Lophotrichous means it's a whole tuft of flagella coming from one pole. Or amphitrichous, flagella at both poles of the cell. So you can see a picture here, paratrichous, you have flagella all covering the bacterium. Monotrichous, here's one just right at one end here. Here's a tuft, so this would be lophotrichous here. And then here's one that shows that amphitrichous where they have a flagella on both ends. Now the main job of a flagella is to aid in motility. In other words, ability to move. So it aids in movement. And it's made up of a filament, which is the long outermost region that actually is the kind of like whip-like tail almost. The hook, which is made up of proteins, and that's attached to the filament, and then the basal body. And this is what anchors the flagellum to the cell wall and cell membrane of the bacterium. So these three parts here are what make up the whole flagella. And how it moves is by rotating it from that basal body. It kind of rotates and helps move that bacterium around. And so it shows you here, since gram-positive and gram-negative bacterium have slightly different cell walls, how that basal body is going to get hooked in is a little bit different. And you can see here the gram-negative has an extra outer layer that the membrane that the flagellum has to kind of be hooked through to get to that inner plasma membrane layer. Now, how a flagella helps the bacterium move is it basically is moving toward or away from a stimulus or a taxis. So if something acts as a chemotaxis, it means it's going to allow the movement of that flagella to make that bacterium move toward or away from chemicals. If it's phototaxis, it means it's moving toward or away from light, because photo means light. And flagella proteins are H antigens. Okay, these help distinguish among variation of species. Remember, E. coli is O157H7. Okay, so those particular proteins that make up flagella are actually H antigens that can label that microbe. And in this case, this is the bad kind of E. coli that gives you food poisoning. And basically, you'd be running to the bathroom. It probably would be coming out both ends, so not pleasant. Axial filaments aid in movement in spirochetes. These are also called endoflagella. They're anchored at one end and they spiral all around the cell. And so you can see it, here it is, it's like all wrapped around. That axial filament is all wrapped around the spirochete. And it rotates, and that's what causes the cell to move, is the rotation of those axial filaments. The fimbriae are short hair-like appendages. They can be at the poles or they can cover the surface of the cell. Here you can see there's short little fimbriae all around this microbe here. Okay, they just look short and hair-like. They're used for attachment to surfaces. So it might help bacteria attach to other bacteria. It might help in forming biofilms, where you get a whole kind of conglomerate and group of microbes together. And it helps with attachment to epithelial cells. So specifically in your intestines, whether it be good bacteria or harmful bacteria that are causing you some intestinal issues, these microbes can attach using those fimbriae. Another structure is the pili. The pili are a lot longer than fimbriae, and usually there's only one, maybe two. They can aid in motility, but here's the big thing they do. They aid in the transfer of DNA, called conjugation. That's one of the ways that DNA can be transferred from one organism to another, is through this, what's called the sex pilus here. And so conjugation is basically the equivalent of bacterial sex. And so that's what that aids in. So that kind of covers all of those kind of external structures. Let me just, hopefully I'm not making you sick by doing this. I want to get back to that original picture here. So far we've covered the pilus. That's this longer structure here, right here. The capsule, again, that's an example of a glycocalyx. If it was a slime layer, that would be another example of a glycocalyx. We've talked about the flagella here and the fimbriae. Okay, those are those short hair-like appendages. Okay, what we're going to talk about in the next section of notes is very specifically the cell wall and the plasma membrane, because those are very important as well.